Yeah.
Okay. Good afternoon, ma'am. Good afternoon, everyone. I'm going to take on this topic, drug absorption from GIT, that is permeability in vitro, in situ, and in-VIVO methods. First, we'll go for the definition of absorption. Uh, uh, absorption is defined as a process of movement of unchained drug from the site of administration to the systemic circulation. And in this method, there are three types, that is in vitro, in vivo, and in situ method. First, we'll go for the in vitro method. What is the mean of in vitro method? In this in vitro method, it means that which carried out outside of the body and which are used to determine the permeability of a drug by using the live tissue of the animal. And in this in vitro models, which have been introduced, the major factors which involve in the absorption process and predict the rate and extent of a drug absorption. And here it is also mentioned that while taking the intestine, that lower experimental, like animals such as rat, guinea pig, rabbits are taken for this study. And then in this in vitro methods, there are some of the methods are including the first one, this physiochemical methods. First one is partition coefficient, artificial membranes, chromatographic retention, brush border membrane, isolated intestinal, and then tissue technique. In number 6.1, tissue technique, it is included four types: everted small intestinal sac technique, everted sac modification, circulation technique, everted intestinal ring or slice technique. 7.1 diffusion cells method or cell culture method. First, we'll go for the partition coefficient. In this partition coefficient, it's also called as a log P, which is in between the oil and water phase. The oil and water phase. And here it is the easiest property which can be determine the drug molecules. And it is also to provide the measure of the lipophilicity of the molecule. And it can also use to predict the extent that will cross through the biological membrane. Some example like octanol is taken here. This octanol is also oil phase, which have similar properties to biological membranes. And this log P, it is important that it does not take the degree of ionization into consideration, and hence log D is used. Here log D and log P is one difference is there. In log P, it does not take the degree of ionization into consideration, but in log D, the distribution coefficient, it is it's taken, and thus it take into account the ionization of the molecules. And this is log D, which is better idea extent of drug permeability across the GIT membrane compared to the log P. And then artificial membrane, second one, ma'am, artificial membrane. In this artificial membrane, second one, ma'am, artificial membrane, or it is also called as a synthetic membrane. In this, it is created the membrane which is usually intended for the separation purpose which are used in the laboratory or the industries, or it is also used maybe in a small scale industrial or a mid large scale industries, and it is used in a wide variety of synthetic membrane known. And this can be produced in both organic and inorganic materials, and it is commercialized utilized synthetic membrane which are used to separate in industries and made of polymeric structure, and they can be classified based on their surface chemistry, bulk structure, morphology, and production method. And the chemical and physical properties of synthetic membrane are separated particle as well as choice of drying force defined as a part membrane separation process. And this artificial membrane, the best known synthetic membrane which are include like water purification or reverse osmosis dehydrogenation of natural gas, or by microfiltration, ultrafiltration, or from removal of microorganism from dairy product and dialysis. And we'll go to the third one, that is chromatographic retention index. Here we can take in gas chromatography, this Covert's retention index, or it can call as a shorter Covert's index or retention index, or plural retention index. This is used to convert the retention times into system-independent constant. And in this index, it's named after the Hungarian, which born Swiss chemist, Erwin Kovács, who outlined this concept during the 1950s while performing the research into the composition of the essential oil. And this retention index, which are certain chemical compounds which normalize the retention time of adjacent eluting and alkane. This retention time, which vary with the individual chromatographic system. Example is taking like regard to column length, film thickness, diameter, carrier gas velocity, and pressure and void time. And this derive retention index, which are quite independent of this parameter and allow comparing the values which measure by different analytical laboratories under very condition. And then this retention index, which can help to identify the components by comparing, measure the retention index with the tabulated values. And then the fourth one is brush border membrane vesicles. In this brush border membrane vesicle, it is also a striated border or a brush border membrane. In this is it's like a round shaped microvilli. This microvilli, which cover of the, which have a tight junction between them, and in this brush border, it is cover surface of simple cuboidal epithelial and simple column epithelium, which is found in different parts of the body. And this single microvilli are approximately 100 nanometers in diameter, and they are varies from and their diameter varies from approximately 100 to 2000 nanometer in length. Because of their individual microvilli are very small, so are tightly packed in a brush border. Individual microvilli can also be resolved using electron microscope. And then the fifth one, using isolated intestinal cells. Here, taking a small intestine and perfuse it with enzyme solution, and that release and then are treated with chelating agent or enzyme. And then after that, taking a fresh isolated cells, which are suspended into a buffer solution. And this, when we did it at the time of experiment, the cells are separated and resuspended in buffer, which containing the drug under oxygen, carbon dioxide, and then shaken well. And then after a specific period of time, the cells will separated by filtration or extracted, and the drug absorbed is determined. And then in tissue technique, here in tissue technique, there are four types. Number one is this everted small intestinal sac technique. In this method, which involve a small segment of the intestine, in the laboratory of a laboratory animal such as rat, inverting the intestine and filling sac with a small volume of drug-free buffer solution. And here, both two segments are taken and then are tied off, and then the sac is immersed in an early Meyer flask, which containing a large volume of buffer solution that contain the drugs. Here, the two segments are taken, and then it is tied together, and then it is immersed in the early Meyer flask containing a large volume of buffer solution. And then this flask contents are they oxygenated, and the whole preparation is maintained at the 37 degrees Celsius and shaken mildly. And then at a predetermined time intervals, this sac will be removed, and then the concentration of the drug in the serum cell fluid is determined, and then the assay for the drug content. And then the second one, that is everted sac modification. Here in this method, here the animal is fasted for the period of time like 20 to 24 hours, and water is allowed, and then the animal is killed, and then the entire small intestine is everted. Here the segment, it is 5 to 15 centimeter length. It is cut to 5 to 15 centimeter length at a specific region of the intestine. And then after cutting, then the distal end of the segment, it is tied. Both end, it is then tied, and then the proximal end, it is then attached to the cannula. After that, the segment is suspending in a mucosal solution which contains the drugs. And after that, a drug-free buffer is then placed in the serocell compartment. And then that is for the everted sac modification. And then the third one, that is the circulation technique. In this circulation technique, here also small intestine is taken, but here it is not everted. Here it is involved isolating either the entire small intestine of small lab animal or a segment circulating oxygenated buffer containing a drug through the lumen. Here the drug-free buffer is circulated on the serosal side of the intestinal membrane and oxygenated. And here absorption rate from the lumen to the outer solution are determined by sampling both the fluid circulating through the lumen and the outside. And the fourth one, that is everted intestinal rig or slice technique. Here in this technique, here the entire intestine, small intestine, it is isolated from the fasted experiment, like the animal which have fasted, which is taken from that, and it is washed with saline solution and then dried by blotting with filter paper. After drying, the segment, it is tied at one end and by placing on the glass rod, and then it is carefully everted and cut into a small ring. After it's finished cutting into small ring, and then the everted intestinal ring are then incubated in the drug containing the buffer, and then it is maintained at 37 degrees Celsius with constant oxygenation. After that, under optimal condition, the ring remained viable for up to 2 hours. And then the transport of drug, it is stopped by rinsing the ring with ice cold buffer, and then after that it is drying them. That is for the everted intestinal ring or slice technique. And then the 7-1 of the in vitro method is diffusion cells method. In this method also, small segment or small intestine it is taken, but here it is mounted between two glass chamber which is filled with the buffer at 37 degrees Celsius. And here diffusion of cell containing of two compartment. In diffusion cells method, it contains two compartments, the donor compartment and the receptor compartment. In the donor compartment, which contain a drug solution, and the lower end of which contain the synthetic or the natural GI membranes that interface with the receptor compartment. And in the receptor compartment, which contain the buffer solution. And then the 8-1, that is the cell culture technique. Here, cell culture technique, this is the cell growing, which are under control condition, which are generally outside their natural environment. Here in this technique, the differentiated cells of the intestine originating from calcium carbonate cells or carcinoma of colon, which are placed on the synthetic polycarbonated membrane previously treated with an appropriate metal such as
cells. So type.
Okay. And then and in these models are based on the assumption that passage of the drugs across the intestinal epithelium is the main barrier of the drug to reach the circulation. And then the second method, in-VIVO method. In this in-VIVO method, it is having two types, direct method and indirect method. This in-VIVO method, it is given us the idea about some important factors which influence the motility and the effects of the drug on the GIT can be determined. This in-VIVO, it is different from in vitro and in situ, because in vitro and in situ technique which give the idea about the absorption. And there are two types in the in-VIVO method, that is direct method and indirect method. And in direct method, the first one, here the drug levels, direct method, here the drug levels are in the blood or urine, which determine the function of time. For this, suitable, sensitive, reproducible analytical procedure should be developed to determine the drug in the biological fluid. In this method, a blind urine or a blood sample is taken from the test of animal before the experiment, and then the test dosage form is then administered to the animal, and at appropriate intervals of time, the blood or the urine sample are collected. After collecting, and then the assay for the drug content will start, and then from this data, we can determine the rate and extent of the drug absorption. And in this method, the experimental animal which choose should bear some resemblance to man. Example like pigs. Pigs are very close to man, but they are not used because of it's difficult for handling. And the other animal which are used like dogs, rabbits, and rat. And the second method, that is indirect method. In this indirect method, which measure of the drug concentration in the blood or urine, it is difficult or not possible, but a sensitive method is available to test the activity. Then the absorption study can be done by this indirect method. In this method, pharmacological response of the drug is related to the amount of drug in the body, or the response is determined after the administration of the test dosage form. Example like LD50 is taken. This LD50, this lethal dose, it is used to measure the short term in poisoning potential material, and this is appeared to be dependent on the rate of the absorbing of the drug. And then the third method, that is in situ method. In this in situ method, it stimulates the in vivo condition for drug absorption, which are based on perfusion of the segment of GIT by drug solution and determination of the amount of drugs diffused through it. In this in situ refers to those methods in which the animal blood supply remain intact, in which the rate of absorption determined from these methods may more be realistic than the determined from the in vitro technique. And these models, which are powerfully tools to study the mechanistic aspect of this important process, and this act as a bridge between the in vivo and in vitro method. This in situ method, it acts as a bridge in between in vivo and in vitro method. And then in this in situ method, in that also there are two types. One is absorption of the drug from the small intestine. First one, absorption of the drug from the small intestine. Here also subdivided perfusion technique. Under perfusion technique, Dolostio method, single pass perfusion method, and the second one, intestinal loop technique. We'll go for the first one, perfusion technique. Technique method. Here, in this technique, adult male rats are fasted for 16 to 24 hours before the experiment. After that, a midline abdominal incision is made, and then the intestinal segment to be perfused is then identified, and then a L-shaped glass inlet is taken, and then cannula is secure into the segment, and then the outlet cannula is placed approximately 15 to 50 centimeter from the inlet cannula. These are secured with sutures, and the intestine is replaced in the abdominal cavity, and it is then dissect the rat, and then are connected by means of tubing of syringe capacity of, and then 10 to 30 ml. And then after washing the intestinal segment with normal saline, the syringe is filled with a solution of drug which is delivered to the intestinal segment, and then it is then collected in second syringe and analyzed for the drug content. And then analyze the drug content. And then the second one, that is a single pass perfusion. In this technique, the drug solution which pass through the intestinal segment just one time. This technique is superior to Dolostio method, and in this precise adjustment of hydrodynamic conditions that can influence the blood circulation and put stress on intestinal wall can be controlled. And then second one, that is intestinal loop technique. Here, single or multiple intestinal loop which are used for the study of the absorption. Here also the other male rat are fasted before the experiment, which under the anesthesia, and abdominal midline incision is made, and the small intestine is exposed, and then a proximal ligature is placed around the intestine from the pylorus, and then a distal ligature is secured at a distance of 4 distal to the proximal ligature, and then the drug solution is then introduced into the lumen of the loop by the means of syringe, which is secured by the proximal ligature. And then after the injection, The needle is then removed, and then the proximal ligature is then tightened, and then the loop is replaced into the abdominal cavity, and the incision is closed. And after a specific time, after that process done, then after a specific time, the animal is then sacrificed, and then the intestinal loop is rapidly exercised and then homogenized, and the amount of unabsorbed drug unabsorbed, and then it is then determined. And then the second step of in situ method. Here also, fasted adult male rat is taken and then anesthetized, and then stomach is exposed and the cardiac end is ligated. Here also, an incision is made in the pylorus, in which the cannula is introduced and ligated, and the lumen is washed several times with saline and then subsequently with 0.1 NaCl solution containing 0.15 m sodium chloride, and then the drug solution of known concentration is then introduced into the stomach, and after 1 hour of time, then the solution is removed from the gastric pouch, and then the assay for the drug content. After that, assay for the drug content. Only that. Thank you, ma'am.
Yes, thank you, Sabita, for the good seminar. So we were discussing about the various in vitro, in situ, and in vivo methods to determine how much of the drug has absorbed when you give it, when you administer the drug, how much of the drug has, what is the permeability. You are checking the absorption of the drug. How can you determine the absorption? By using these kind of in vitro, in vivo, and in situ method. So how are the three methods different? What is special in vitro method? So though the name is in vitro, we are used it, of course, in vitro means outside the body, and we are using the live animal tissues. In the we have learned about in vitro dissolution studies. Here we are learning about the in vitro methods to determine the absorption of the drug from the GIT. So we are going to determine the permeability of the drug by using the live tissues. So why we use these kind of in vitro models? So you can assess or find out the major factors involved in the absorption process and predict the rate and extent of drug absorption. Okay, so the intestine of lower experimental animals are used: rats, guinea pigs, rabbits. These are usually selected for the study. So we know that these are close to the human models. That's why we select the guinea pigs, rats, and rabbits for the study. So what are the different in vitro methods that were determined? You can use some physicochemical methods where you can determine the partition coefficient. Also, you can use artificial membranes. They may be made from organic or inorganic matter, and you can also use chromatographic retention indices to find the extent of absorption, permeability. You can use the brush border membrane vesicles, isolated intestinal cells. Then you can use various tissue techniques which have been discussed: everted small intestinal sac technique, circulation technique, everted intestinal ring or slice technique, diffusion cell method, cell culture techniques. So what is important, what we are trying to find here, if you use this partition coefficient, you can know how the drug has, it's a very important property of the drug molecule. Okay, the partition coefficient, you can know the measure of lipophilicity of the drug. You know that lipophilic drug, non-ionized, non-polar lipophilic drugs can easily pass through the biological membrane. So you can find out the extent of permeation. If you know, you can measure the of the lipophilicity of the drug molecule can be predicted. To what extent that particular drug can cross the biological membrane. You can, if you assess using the partition coefficient. So what is the partition coefficient? It is partitioning of the drug between the oil and water phase. So you can denote, you can find the log P value. And one of the main octanol is mainly used as the oil. Octanol is used as the oil phase, as it has similar properties to the biological membrane. So in partition coefficient, there are two terms. One is log P, another one is log D. Log D is the distribution coefficient, where the aqueous phase is at a particular pH. So it takes into account ionization of the molecule at that pH. So the log D, which is measured at intestinal pH, will give a better idea about the extent of drug permeability when compared to log P, because the drug may also be in the log D alone takes into account the degree of ionization, while log P only does not consider the degree of ionization. Of course, it is the unionized drug which goes through the permeates easily, but the log D will give a better idea about the extent of drug permeability. So various artificial membranes or synthetic membranes may also be used for this absorption studies. They may be made from organic or inorganic materials. And there are also chromatographic retention indices. You know that each drug will have a particular retention time or retention factor. If you know those RF values, you can detect the drug that has. You can tables of retention indices can be used to identify the peaks in the drugs when you measure the retention indices. So another thing is a brush border membrane vesicles, then isolated intestinal cells. Here what is done is the small intestine is perfused with enzyme solutions which release the cells that are treated with circulating agents. Okay, so in this, what is done, the isolated cells are suspended in the buffer solution, and after the specific period of time, you are removing the cells, separating the cells and extracting and determining how much drug has been absorbed. So you may use isolated intestinal cells. You may also use tissue techniques. These are quite familiar with you, the everted small intestinal sac technique. And then there is the modified technique. So these segments or the intestinal segments will be suspended in mucosal solutions containing the drug, and you can determine the, then also there are several other techniques like the circulation techniques, then the everted intestinal ring or slice technique, diffusion cell method, which is quite familiar to you, the donor compartment, using the, you are mounting the segment of the small intestine between the two glass chambers, which is filled with buffer. So the diffusion cell, you would have learned about the Frank diffusion cell, and you are using it in your lab also. So it has both the donor and receptor compartments. The donor compartment usually contains the drug solution, and then the lower end there will be the membrane at the interface of the receptor compartment, and the receptor compartment will have the buffer solution. So you can at different time intervals, you will be withdrawing the buffer solution and noting how much of the drug is available, has diffused or permeated. Similarly, cell culture techniques can also be used, where you are growing the cell under controlled conditions. You would have heard about CACO2 cells. They are the cells of for the used for the tissue culture. Usually, for cell culture techniques, we use these cells of carcinoma of the colon may be used. And here, these models are based on the assumption that passage of drug through the intestinal epithelium is the main barrier for drugs to reach the circulation. So you are assuming that the intestine is the main barrier for drugs to reach the circulation. That's why you are adapting such kind of techniques to check the permeation of the drug through the intestine or through the GIT in general. And in vivo methods, there are three types of methods: in vitro, in vivo, and in situ methods, which you may use to know how much drug has been absorbed from the GIT. You know, absorption is very important, so that only then, once the drug reaches the systemic circulation, it can give its therapeutic effect. So, in methods, you have direct as well as indirect methods. So, in direct method, what is done, the drug levels in the blood, that means the plasma drug concentration is determined, or how much drug has been excreted and changed through the urine. That can also give an idea about the extent of absorption, or what is the bioavailability, how much drug has reached the systemic circulation. So in this indirect method, for if you need to know the plasma drug concentration, you need to have proper bioanalytical techniques. You need to have suitable, sensitive, reproducible analytical procedures so that you can determine. So what is done, you are taking the blank urine or blood sample before, and then you are giving the test dose to the animal, and at appropriate time intervals you are collecting the blood or urine samples and assaying it for the drug content. You can use HPLC methods where you can get the peaks, and you can find the AUC. You can thus determine the rate and extent of absorption by these kind of direct methods. So usually animals like beagle dogs, albino rabbits, and mice, they are used for these kind of animal studies. And in the indirect method, what we do is, when you cannot when there are no proper bioanalytical techniques or for measuring the direct drug concentration in the blood or urine sample, you have to use indirect methods where the pharmacological response is related to the amount of that is measured. The pharmacological response is measured after administering the test dosage, and you see the LD50 is dependent on the rate of absorption of the drug. Then there are also in situ methods, which are a bridge between the in vivo and in vitro method. It is simulating the in vivo conditions for drug absorption. So it is based on the perfusion of a segment of the GIT by the drug solution, and is determination of the amount of drug which has diffused. So these models are powerful tools to study the absorption process. So you can learn the absorption from the small intestine by these methods, perfusion techniques and intestinal loop techniques. These are the in situ methods which are used. In perfusion techniques, there are the Lolusio method and the single pass perfusion method. There is also the intestinal loop technique. So we discuss these things in detail. So let's go through this topic once again and make a note on the drug, the GIT. What are the different methods to determine the permeability: in vitro, in situ, and in vivo methods. Okay. In vitro methods.